I'll just ask Leandro to start sharing and get started with this.
Well, I think it's good that these two talks are one next to each other, because they are going to be quite connected, and then we can chat here or in the Discord channel. So let me present myself. I am Leandro Radusky. I have a degree in computer science, but then I moved to the fields of bioinformatics. I am finishing a PhD in the CRC in Barcelona, and starting working in managed tech to work in the first steps of the biocrystal library, among other projects. And today I will talk about these first steps of the crystal bioinformatic ecosystem, what is there, what is not, and the idea is as well trying to convince you why Crystal is a good choice for your bioinformatic projects. And one thing that I don't really know is how many bioinformaticians are already in this room, or how many of them will see this talk afterwards from the recordings. So in a way, I will make a presentation for those that are not too familiar with the bioinformatics fields, and as well, I am talking more specifically to bioinformaticians to talk about crystal benefits. But just to make a small introduction, even if computer processing and from the very first steps of computer science, computers were made for numerical analysis, we can say that the bioinformatics fields has its departing point from the analysis of the double helix and the determination of the double helix of DNA. And because in this very moment, we start to interpret experimental data in order to arrive to new biological knowledge. Here I am just pointing out some milestones of coding that are important to the bioinformatic fields. And the thing that I want to point out here is that you can see that we started with numerical analysis, but soon there were other things that bioinformaticians do, as Peter was mentioning. In 1965 and 1971, we have the birth of the biological databases and the field of bioinformation, which is a whole field within the bioinformatics. You can have a huge field within bioinformatics, which is meant to do images analysis, or you have sequence analysis, which is another kind of analysis which has to do more with algorithms that with a specific numerical analysis, and is related to what Peter was talking. But we didn't have, let's say, a proper and established field of bioinformatics until the explosion of the next generation sequencing and the availability of the huge amount of data that genomic sequencing provided to us. The amount of data started to grow exponentially, and the bioinformatics became a data-intensive established field within sciences. Even though when I was finishing my master degrees in computer science, and I want to move forward because I was interested in bioinformatics and biological problems, in the lab that I started to work, which is a lab that were doing more, where they were doing more biology, and they were not specific persons doing bioinformatics. This was what was considered bioinformatics in 2012 when I started in the field. So just to mention what and to extend a little bit what Peter was saying about the different profiles of what an bioinformatician can do. For example, you can have persons that are running atomistic simulations that are programmed in C++, and they have to run in clusters or in GPUs, and then they do data processing in Python and R, as Peter was mentioning, or they are doing images analysis, as I was mentioning before, and generating the final graphs for the papers in more statistically friendly languages like Python or Excel, or they are searching to compound database to look into proteins to search for new molecules, and they have to have a strong knowledge within the databases, or they are just doing experimental work and they want to interpret the data that they are obtaining to generate meaningful information and run small statistical analysis, or they are processing omics data, which is a huge amount of data, and they want to make this data available for the public in public databases, and they are mounting websites with this. And these are the technologies that I'm putting just because I know people that are working on them, but this is of course just a small cut of all the things, all the different things that are considered in bioinformatics nowadays. So as Peter was mentioning a little bit, what do we have in bioinformatics? If we want to know which is the state of situation of bioinformatics, I think that a good approach is to see the shows that the bioinformatician laugh about. Basically, a bioinformatician is always struggling between the domain knowledge and the coding skills, and you have a lot of people that, as Peter was mentioning, that is not coming from computer science or engineering, but from biology or other fields, that in the moment that they have to learn, if the language is not friendly, they are struggling, and also with the statistical part and computer science specific features. You have a lot of competing standards or things or different things that are available in different formats through different formats, and they are needed to be processed in different ways, or you can just be like this guy, which is absolutely desperate to know which bioinformatics could be. From an educational point of view, we can say that in 2014 there was this paper, but there was also a lot of starting degrees of bioinformatics in 2001. This is just a summer camp of bioinformatics, but there were not bioinformatics degrees. But even in 2014, there were no core competences definition of what a bioinformatician should do, and in my opinion is because this is of this wideness of profiles that you have within the fields. If you take a look of the kind of resources that are there and how they grow through the years you can see that in the last 10 years there was an explosion of people working in bioinformatics, because there is as well an explosion, as I was mentioning before, in the amount of data that has to be analyzed, that has to be and could be only done by computers, and because of these different profiles that are coming to bioinformatics, what you have in the last 10 years is an explosion of interpreted languages, especially Python and R, because they are let's say easy to understand and easy to start programming with for people which is speaking in math languages and has not a specific computer science requirement knowledge to be programmed. But as well, you can see that C++ or Sharp or C are still alive, because there is a need as well to perform on tools, as Peter was mentioning in the previous talk. There is things that you want to do fast, or a small analysis that anyone can do to make a small statistical interpretations. But when you want to really work with data in a performant way, you have to rely on languages like C++ or C that will allow you to have a good performance. So basically, this is the state of the situation. This is where we are, and in the A side of the bioinformatics ecosystem, you have, for example, in these two languages that I was mentioning, you have in Python a great ecosystem of things that that are available for you to perform analysis. The same is happening with R, which is a language which is more math-like language meant specifically for statistical analysis. But in the B side of this, you have first of all that these languages has a lot of libraries that are called libraries for analysis and interpretation that are not self-hosted, let's say, that are not programming in the very language that they are proposed to, or you have R, which is not self-hosted as well. And this is not because self-hosting, because of the self-hosting, but you have people which is working on a specific scientific problem and maybe starting by some analysis, but then afterwards really goes deep into the problem and needs to know what is there. And when you have a library which is implemented in another language, then you are putting a value for the people which is going deeper and deeper in the problem that they are attacking. And this is something that Crystal could be to be a superation of this. Another problem that you have is that, for example, in bioinformatics, a lot of things that you do is to develop pipelines that you start with raw data from some experiments, and then you are plugging in different analysis tools that are transforming the data through several steps, and these different tools are programmed in different languages or with different versions, or etc. And even though these three tools are super cool and I love them, of course, this for our informatician that doesn't come from the computer science field or is just a biologist trying to perform some analysis could become a little bit complicated and messy how to not degrade your environment. So in terms of this is more or less what was happening in the past 10 years in bioinformatics. We have this explosion of data. We have this necessity of a lot of people analyzing this data, and these two languages, R and Python, that are super friendly for scientists, which allow a lot of people to enter into the field in bioinformatics, both in academia and in the industry. The thing which is happening now is that there is a need for tools that are performant. Again, like Peter, I don't want to make a word of languages or nothing like this. It's just to make a point. For example, in a language like R, which is a more mathematically oriented language, you have a new development which is great, which is Julia, which is also a language which is more mathematical like, and for Python, which is a more general purpose language, and C++, which is a performant language, you can have the best of both worlds in Crystal, and this is what I want to defend in this talk. You can put Ruby here, but in the specific field where I came from, like Python is a little bit more used for these things that Peter was mentioning, because there is more machine learning tools or more bioinformatic analysis ecosystem available tools than Ruby. And I think that in Crystal you can have best of both worlds. Even for a specific numerical analysis, I think that instead of having a code which is more mathematical like, to develop in Crystal is a good idea, but I think that it's in this very same track that in an hour or so you will have another talk defending this argument. So I don't want to go forward into this, but also I think that when you are doing a specific numerical analysis, I think that if you're using a language which has the characteristics of Crystal, you are making it more reproducible, more productive, etc. Just to mention how Crystal performs compared to other languages, I was taking as well the Julian language, which is the benchmark of the Julian language for some operations, and I was comparing Crystal performance of these algorithms that they propose in Julia versus all these other languages. You can see that there is a quite a clear difference between those languages that are compiled and those languages that are interpreted. Also, Crystal has a great performance in large data set handling, which is something which is required a lot in bioinformatics projects. For this, I was taking the best algorithm in the Queryverse benchmark for each one of the three languages, Python, Julia, and R, and comparing it to Crystal. You can see that Crystal is performing very good. And the thing that I am working in now, and the thing that should be essential for having our informatic Crystal ecosystem and bioinformatic Crystal community is a BioCrystal shard, which is basically allowing you to have parsers for the most common data formats and ways to interact with the most common online resources that are there, which is sequences, protein structures, and for example, Peter was mentioning BCF. That there is even if there is a lot of different formats and a lot of different standards, there is for each kind of biological type, you have more or less a standard that you can work with. Basically, what I'm doing here is to run a BLAST, which is a sequence alignment tool, in order to find a sequence homologue in a sequence database. Here, I just made this example to show you how you can interact and make a system call in a memory variable that allows you to generate the database of sequences, and then you just can run the BLAST program and get again the output of this on a variable in memory without passing through files. And because of this, is returning you data in XML type. This is something that I want to show you as well, which is how easy it is to parse an XML file with Crystal, how friendly it is a language in order to make this parse and how it is doing it. Just to show you a more sophisticated example, here I am going to take a big data set. Basically, here we have 25,000 machines, more or less, which are followed along 1.4 thousand cells. So you have a huge dimensional space, and basically you have all these cells in a single RNA-seq experiment. And you want to classify the different cells according to its cell type, which is something that can be done in several manners. But for this, I am going to use neural networks, and specifically the library that I chose was nan.cr, because I know that the next talk is going to introduce another neural network library, so I don't want to overlap, and I will show you how easy it is to do this and how friendly. Here I am just reading the huge CSV and shuffling the data just to read it and have it stored in the machine. Then I am defining the different cell types according to the output of the neural network. I will have 7 categories of cell types that I will use for train. And then basically here I am creating the training variables. Here, for example, this is how I'm normalizing each one of the genes around the variable. You can see that the syntax is very friendly and understandable. And to make the neural network is as well super friendly and easy as can be done with PyTorch or Tensorflow. I know that there is not as many features that in these two very well developed libraries, but you can see that there is a very friendly and familiar way to define the neural network. Basically, here I am putting in the input of the neural network the gene expression for each one of the cells, which I am going to use for training. Then I am defining 7,500 nodes for the hidden layer of the cell of the neural network. And the output is the 7 categories, and then I will use some cells for training and to measure the accuracy of the classifier. As well, and I want to remark on this, Tensorflow or PyTorch, which are very cool, and I love them, and I have used them. Libraries for Python and doing neural network on Python are done basically in C++. So in the moment that you really want to get involved in the development, if you have to learn C++ or basically do something on top of Python, while this library is done mainly in Crystal, except for the Blast calls. And I think this is something which is super cool and working with tensors and super parallelized, etc. Here you can see basically how easily I run a neural network. Basically, I take some data to train the network, and then I move forward this data to the network. But basically, if you know something about neural networks, the third step is to propagate the results to change the internet state of the nodes. And basically, these two things are to evaluate the accuracy of the epoch and to print the results. Here you can see that this is working. Well, this is not properly synchronized, but this is working. This is working in a parallelized and tensorized way. This can be run also on GPUs, and you can see that the accuracies are achieved as any other library, and in a pure crystal manner. So just to remark what is already available. Well, these characteristics of self-hosting that I was trying to stress out. You have a good performance, as we showed with the benchmarking graphs. You have parallelism. You can really easily make portable executables of your project, which is something which is not as easy with other languages, and you have great standard library for dealing with the most common formats. What is happening already? Well, you have linear algebra, tensor matching neural networks, as I was showing you with Node.cl, and you will have the next talk about it. You have ongoing developments of your Crystal data visualization. You have Crystal also running on notebooks. There is a project called like Crystal, but after the Alice talk of before, I think that this is going to move even faster. And you have the BioCrystal library to deal with the most common biological formats ongoing. And what is still missing? Well, a little bit what Peter was mentioning, a wider and consolidated ecosystem. You don't have yet, and this is something that people use a lot, a data frame like handling, which is something which is quite established in the bioinformatics field. And as far as I know, I even found some statistical modeling features, for example, Markov chain of the Cal or things shards to deal with this kind of statistical modeling tools. So, in my opinion, the take-home message is time to pioneer. I think for those coming from the academia, well, you may know that when you pioneer in something and new things, well, they can establish and they can be really useful for the community. And this is something that within science is highly appreciated and rewarded. And with this, I want to thank you. I don't know how long I talked. I hope that it didn't take too much time.
Leandro, you're perfectly on time. We started a bit later than expected, so we're wrapping up just in time. We have 3 minutes for question. I don't know if people want to ask questions right now. Also, a note that this is, Leandro marked the beginning of the scientific track, although we've been talking science with Peter as well. but just also to say that MXNet, for example, which is a library that Leandro has mentioned, is something we'll be talking about in the next talk in 2 minutes time. So I'll actually promote Todd while people start asking questions. And it was great to see NumCR mentioned in your talk, Leandro. Great to see that being put into use. I've played a bit with it myself to do some very, very simple linear algebra operations and found it quite easy to use and very performant. So that's a great place to be. But as you say, there's no such a thing as a standard go-to library at the moment for statistical modeling or even for data handling on the kind of a data frame like sort of approaches. I can see Ian is asking about when we're going to be seeing CRISPR plus Crystal, but I'm not familiar with CRISPR.
CRISPR is a gene editing technology, but in a way the sequence analysis is done. But let's say CRISPR is a pure experimental technique. So I think that things could be developed in terms of sequence analysis or generating things, but it's not something that is happening now. I don't know.
Thank you for asking, Ian.